our presentation on investigating the antimicrobial properties of Aplasia californica ink. My name is Rachel Hennegan, and I am a senior in a biology major.
My name is Kylee Schanbacher, and I am a senior biology and chemistry major.
My name is Kenzie Collier. I'm a senior biology major, and our mentor is Dr. Kimberly Hale.
So the common name for the Aplasia californica is the California sea hare, and they are commonly found off the coast of California in tide pools. When they feel threatened as a defense mechanism, they release a viscous ink that ranges in color from dark purple to bright fuchsia. And this color is a direct result of their diet, which consists of red and brown algae. The toxins found in their ink is also a direct result of their diet. So in these pictures, we show the Aplasia californica that we use in our studies. All there.
So our research group has been studying Aplasia and their ink for a few years now, and we have been able to consistently demonstrate that the ink from our Aplasia contains antimicrobial properties. We can see from this table, though, that we get a great degree of variation in between the different inks. So our goal is to try to figure out what is causing this. So as said before, the main goal of our project is to try to identify the differences in the ink and why there is such a varying degree of variation with the antimicrobial activity. Antimicrobial activity is, in other groups, they have found that this was caused by escapin in the Aplasia ink, but we hope to identify this in our ink as well and also see if there's something else that could be contributing to it.
Ink collection was an important aspect of our research as we needed to collect ink very often in order to run all of our tests. Finding a safe and effective method for collecting the ink became a goal of ours very early on in our research. To learn more about the specific protocols for ink collection and storage, please refer to the poster presentation next week.
Our research consisted of testing the Aplasia ink in 4 different ways to identify these differences in the ink. We did zone of inhibition testing, Western gels, electrophoresis gels, and IR testing. Each of these techniques right here will be explained to you today. So we first began testing the ink from our Aplasia with zone of inhibition testing. We did this to see if we could demonstrate that Aplasia ink truly did have these antimicrobial properties in it. Zone of inhibition testing is a fast, qualitative way to measure the ability of an antimicrobial agent in inhibiting the growth of a microorganism. This is a standard procedure used in hospital laboratories to test the effectiveness of antibiotics. In our research, we used 30 microliters of a bacteria. We used Staphylococcus aureus in our research, and we spread this on a Mueller-Hinton agar plate using a sterile spreader. We then soaked each disc in 40 microliters of the appropriate ink and had them soak for at least 15 minutes. We then placed each disc in the center of a Mueller-Hinton agar plate and incubated them at 37.5 degrees Celsius, lid side down, for 24 to 36 hours. We then measured the zone of inhibition. We do this by going from the edge of the disc to the nearest area of bacterial growth. The larger the zone of inhibition, the greater the antimicrobial activity is. So this is the first trial that we ran. We have some representative data to show you. So as said before, the zone of inhibition is measured from the edge of the disc to the nearest area of growth. So looking at B2, these are both B2, they appear to have pretty large zone of inhibitions. But if you look closely, there is a circle of bacteria surrounding the disc, about 2 to 3 millimeters away, making the zone of inhibitions very small. So we do see that B2 does have a zone of inhibition, though, meaning that it does have antimicrobial activity. We also can see the ability of 2 antibiotics, tetracycline and penicillin, in inhibiting the growth of this bacteria as well. And we can see that B1 does have similar abilities to tetracycline. So this is the second trial that we ran. In this trial, we can see that there is quite the variation between inks, and even like the same ink collected from the same aplasia, but just collected at different times. And we want to know why this is. It could be related to how much water was collected with the ink, or just specific differences in the ink secreted from the aplasia at that time. This is why we began testing the ink in more ways, such as the gel electrophoresis and Western gels, to try to get to the bottom of this variation. So looking specifically at A1, B1, C2, and C3, we can see that using the same samples collected from the same aplasia at the same time, that we get the same results, meaning that our variation isn't due to a procedural error, it's just due to the variations in the inks collected at different times. So we wanted to see that if we spun down or centrifuged the ink to get a pellet of ink in the supernatant and then compared them, if we would get less variation between inks due to removing the excess water from the pellet. We would most likely expect that the pellet of ink would have a higher zone of inhibition than the supernatant due to the supernatant being mostly water. We are still in the beginning of perfecting this protocol and testing this theory, so we don't have complete steady results yet, but we can see that the B pellet has a much higher zone of inhibition than the B supernatant, but we can also see that this is not the case in the A pellet and A supernatant.
Gel electrophoresis is a process that uses electricity to separate molecules based on their size and charge. An electric current is used to move the molecules through a 1.5% agarose gel. We use gel electrophoresis in our lab so that we can separate these molecules based on their size and charge. To load a gel, pipette 30 microliters into their corresponding well, fill the box up, lightly covering the gel with 1x running buffer, and then run the gel between 100 to 120 millivolts for a desired time. The wells of the gel start from the lower left side and work their way to the right. You can see that in wells 3 and 4, 10 and 11, that we got colored banding on the gel, showing that there is significant interest in Aplasia G's ink. The color banding on the gel shows purple and brown colored banding, which shows that there is there's differences within the ink that account for the different banding. In this trial, we also tested the poo of the Aplasia to see if there was any antimicrobial activity in it. Unfortunately, in this trial, the poo did not create any colored banding on the gel. For this trial, we filled the wells with 30 microliters of sample and ran at 150 millivolts. Once again, in this trial, we see that in wells 4, 5, 12, and 13, we get the only colored banding on the gel, once again showing that there is significant interest in Aplasia G's ink. This gel shows out how the same ink collected from the same Aplasia can have different colored banding or no banding at all, as we tested G1, G2, G3, and G4, and only G1 and G2 created banding. In this trial, we focused on 3 different Aplasia not used in the first 2 trials. We wanted to see if we could recreate the same colored banding in all of the Aplasia. In this trial, we found colored banding in wells 1, 6, 10, and 15, which corresponds to A1 and C1. This shows that inks A1 and C1 contain the same colored components as G1 and G2 and create the same colored banding as the previous trials. In this trial, we used new techniques with the ink. We used centrifugation to see if we could get different results thinning down the inks to use the different components of it. Centrifugation is the technique used in the separation of heterogeneous colloidal substances. Ink particles are colloidal in nature. In centrifugation, by the application of centrifugal force, the colored components of the ink get separated. At the end of the centrifugation, you have a supernatant top portion and what is known as a pellet on the bottom portion. This technique allows us to test the different properties of the ink to see which component creates the colored banding on the gel. In this trial, we ran 30 microliters of sample at 120 millivolts for 1.5 hours. As you can see, this gel ran very light, which we believe to be due to too much water used during the collection of the ink. It also accounted for a small amount of pellet gathered after centrifugation, which made it very hard for us to pipette the pellet into the gel. Looking at the gel, you can see that there is light colored banding in wells 1, 2, 5, 6, 9, and 10. These results tell us that in all 3 of the Aplasia tested, the plain ink and the supernatant only were the only ones to create colored banding, and the pellet of each created no banding. Based off the findings from the previous trial, we looked to run another gel specifically looking at the plain ink and supernatant only of Aplasia D and F and the pellet of Aplasia H only. The wells were once again filled with 30 microliters of sample and ran at 100 millivolts. This gel did not run the way we expected it to, and we only got colored banding in well 9 that contained the ink pellet from Aplasia H. Seeing this was very confusing to us, as from the previous trial we got no colored banding in the pellet at all. But this is significant that Aplasia H's pellet did create colored banding. We believe this is possibly due because after centrifugation we did get a very large pellet from Aplasia H, which made it much easier to load into the gel, which we believe would be the cause for this banding. Now we look to compare the zone of inhibitions with the gel electrophoresis. This gel and zone of inhibition show the direct correlation between Aplasia G's antimicrobial activity and its relation to the color banding it created. In the gel, we see that the only significant banding, compared with the zone of inhibition, shows that the plates G1 and G2 of Aplasia's ink had significant effects on inhibiting the microbial growth of Staphylococcus aureus. This is exactly what we were looking and hoping to find through this research.
So looking at this table, we can see that G1 ink and G2 ink have much higher zone of inhibitions than the rest of the inks. And we can also see that looking at the gel, that G1 and G2 on both of those are the only ones that have this large purple banding. So we hope to do further studies on how this purple band relates to the antimicrobial activity.
This was another gel and zone of inhibition that was run using the same inks as the first time. We wanted to run the same inks again to see if we would get the same or similar results and to rule out any errors from the previous trial. In this trial, we ended up getting even better results, as the gel was able to run longer and we could see the banding on the entirety of the plate. Aplasia G's ink was by far the most interesting and gave the best results throughout the research. Knowing this, we looked to do even more further research on all the Aplasia's inks.
Although this is a different trial, we still got similar results. As we can see, the inks G1 and G2 still had much higher zone of inhibitions than the rest of the inks. And again, looking at the gels, they were the only ones that had this purple banding. So since we knew that this purple banding most likely correlated with why G1 and G2 had higher zone of inhibitions and more antimicrobial activity, we decided to cut these bands out of the gel and use them for their own zone of inhibition study to see if that specific component that the gel was picking up was what was responsible for the antimicrobial activity. We are still trying to perfect this protocol, so we don't have study results yet, but we are trying to look into this further.
So a Western gel is used to separate proteins by their size in a polyacrylamide gel. And so we use a ladder in the first and the last well of the gel that has known protein sizes so that we can compare the bands that we get from our inks to the ladder. So our goal with using the Western gel is to be able to identify the protein that may be responsible for the antimicrobial properties of the ink. You can see in this picture here, we have inks loaded into the wells, and you can see the variation in color of the inks. And so after we would load it like this, we would run this at 200 volts, and we would stain it to be able to see the bands, the protein bands. And this procedure is really difficult and precise, so it did take our research group a little bit of time to produce Western gels that could be used for data purposes. So this is a Western gel that was performed on March 1st, and this has significant data because you can see that all of the inks had a protein band that came up at 50 kilodaltons. So if you look at the gel, there are some faint bands right here. Oh yeah, there we go. There are some faint bands right here at 50 kilodaltons all the way across. And these are the protein bands that we're looking at, while this is just the ink running down the gel. So you can see we ran this gel about halfway. And then these are the ladders here and here. So we used these same inks in a zone of inhibition study on the 3rd. And you can see that all of these inks had a zone of inhibition of 5 millimeters or greater. So that's a pretty significant zone of inhibition. This is a Western gel that we did on the 23rd of March. And the only inks that we got this banding, oh my gosh. The only inks that we got this banding at 50 kilodaltons were right here. And those are the D whole ink, the D supernatant, and the spun down D pellet. So in a zone of inhibition done with these same ink samples on March 22nd, we can see that these same D ink samples all had the highest zone of inhibition. So we are consistently getting protein bands at 50 kilodaltons for inks that also have really high zone of inhibitions. So we are hoping to keep comparing our zone of inhibitions to Western gels so that we can identify the possible protein that might be responsible for the antimicrobial activity. We've also just started performing IR on our ink samples, and since we've just started, we haven't really been able to analyze our results too well. But IR stands for infrared spectroscopy, and it's used to identify the functional groups of molecules. So in these two charts here, we have one for the C whole ink and one for the spun down C pellet, and you can see that they both have different functional groups, even though the pellet was spun down from the C ink. So we have collected a significant amount of data showing that ink collected from various Aplasia californica yield different results on agarose and western gels, and that these results correlate to the measured antimicrobial properties of those inks.
For further future studies, we would like to further investigate the various properties of Aplasia's ink that account for their differences. We would like to examine the various Aplasia's inks and determine the different sizes using the plate reader. We would like to investigate the chemical properties of the Aplasia inks by doing more IR studies and also NMR studies. And lastly, we would like to perfect our centrifugation techniques, as explained earlier, in order to study them better and to separate the components.
We wanted to include a couple pictures of us actually performing the research. We are all so thankful for the opportunity to do this research, and we got to learn so many new things from it.
For our acknowledgements, we would like to thank our entire lab group who worked really hard this semester and the whole year. We'd like to thank Dr. Michelle Combs for help with the Western. We'd like to thank Dr. Deskins for help with the IR, and we'd like to thank Dr. Lee Enger for stepping in while Dr. Hale was out.
So that's definitely something that we're looking at right now. That's kind of what we were talking about when we were talking about spinning it down and centrifuging it, just seeing if we got that excess water out, if we would get like more or less variation in between the inks. We're still just beginning to do that, so we don't have complete study results yet, but it seems like that is the case.
So this has important applications, mostly to the medical field, because you can compare it to, for example, that certain Aplasia's ink was really close to the effects of tetracycline. And in the future, we're hoping that spinning it down and being able to concentrate the ink into, or concentrate the antimicrobial properties, can be further experimented on.
For me personally, I am continuing on to the medical field, specifically radiography. And I just believe that having a background where I can apply skills and research on my own time was a good skill to have, going into a career where I'm going to be very hands-on with equipment and working by myself most of the time. So having that background and knowledge of different aspects of biology and research was very helpful for me, I believe.
So we're hoping that the information gathered in our research can be further applied towards that goal, not necessarily directly from our research, but we're mostly just trying to identify the component of that makes the ink antimicrobial, so that hopefully maybe you could concentrate that and use that in various ways.